A very good morning to all of you. We are here to celebrate today Global Women's Breakfast, and actually we are here to celebrate women today. Our first guest, Dr. Ishu Saraogi from ISA Bengal, she has joined. So I welcome you, ma'am, on this platform.
Thank you,
And yeah, and we basically are celebrating women and girls in science today. And however, a lot have been said about the participation of girls and women in STEM, and only on 11th February we celebrated the day also. But even then, what we feel that the scenario which is very evident to us, or the scenario which let us feel enlightened, that scenario basically belongs to the 2% of the girls, I think, which are in higher education. And out of those 2% girls which are in higher education, basically I think that another 2% of 2%, they have the chance to think about that, what they want to do, how they want to proceed in their career, without thinking about family or without thinking about other things or other issues and other barriers. I would just like to confess over here that yes, this is a man's land, and even today science is like believe that this is a area where there are more opportunities, or where there are more facilities which are available to male as compared to the female scientists. Looking at the industry scenario also, I as a teacher and as a mother feel that when it comes to sending our daughters to the industry, even industry is also not even very susceptible to take these or to consume these girls. But even as a parent, we are also apprehensive that is there everything which is okay there, is this will this be okay to send our girls to the industry when they have to work in odd hours, they have to work in the night shifts or something like that. So this taboo or this lacuna which just compels us to think in a biased way when it comes to girls and boys, it still haunts us, and it still exists in the scenario of India, I can say that. So we just want to talk today about breaking barriers in science. We just want to talk that how this barrier can be broken, how these girls can be taken into the mainstream. So we here in Government Madhav Science College Ujjain are trying not to make our girls thoughtful about, or make them think about all these things, but also to our boys. Today, many of the boys have joined, and rather from 2 days, we were convincing boys whenever we were saying that we are celebrating this day. So the boys were saying that, ma'am, this is Global Women's Breakfast. How come we are a part of it? So we said that no, you are more part of it, because you are the ones who are creating hurdles and problems for women. So you should come forward, whether it's your sister, whether it's your mother, girlfriend, wife, anyone. But being a boy, it's your responsibility to provide a smooth and safer platform to the girl counterparts, whosoever you encounter into. So with this theme in our mind, we are celebrating this Global Women's Breakfast, which is an initiative of IUPAC. And IUPAC is celebrating this Global Women's Breakfast on an international platform. Many organizations today are celebrating this Global Women's Breakfast. Association of Chemistry Teachers in India, Association of Chemistry Teachers is a pioneer of this in India. And on the behest of ACT, we are celebrating this in our college also. There is a support which always exists for us in the form of ACT, because the ACT chairperson, Dr. Brajesh Pare, he happens to be the head of the Department of Chemistry in this college. So we are a privileged one, as we have the chairperson with us. So ma'am, I just want to inform you that this is a college which is A++ grade accredited. NAC accreditation was there in this year only, and we got our grade on July. So we got this A++ grade, which is a very high grade. As in talking about government colleges, we are the we are on the topmost rank in India. So it's a privilege and it's a matter of pride for us. So we want always to mention it that we are A plus. And this is a science college where we majorly practice science. We have research center and we have PG classes, we have UG classes and all that. To be physically with us. So thank you very much for being here. And now I would like to invite Head of the Department of Chemistry and ACT Chairperson, Dr. Brajesh Pare, who is with us to speak a few words. But before that, let me introduce Madam Ishu Saraugi to the audience so that they may be able to comprehend. Dr. Ishu Saraugi, she received her BSc in Chemistry from University of Calcutta and MS in Chemistry from Indian Institute of Science, Bangalore, working with Professor T.N. Guru Rao. In 2008, she obtained her PhD from Yale University under the supervision of Professor Andrew Hamilton, focusing on the synthetic mimicry of protein secondary structures and their application in inhibiting protein-protein interactions. During her postdoctoral stint at Caltech with Professor Sushos Shan, she worked on elucidating the molecular mechanism of biomolecular interactions involved in protein transport. In 2013, Dr. Saraugi joined Indian Institute of Science, Education and Research, Bhopal, where she works on developing chemical tools to modulate biomolecular interactions. Their research group focuses on development of novel antibacterial and anti-amyloidogenic agents. She is a recipient of Ramanujan Fellowship and Power Fellowship from the Science and Engineering Research Board, India. Their work was recently featured in the Chem Bio Talents collections by the Journal of Chem Biology, Willie Easton. So welcome you, ma'am. I welcome you in this Global Women's Breakfast. And I have with me Sneha Purwal. Sneha was communicating with you. She's a young faculty in our college. Thank you, ma'am. Pare sir, I invite you. Please speak with us.
Thank you very much, Professor Kalpana Singh and Professor Ishu. Thank you for coming for this program. United Nations, under IUPAC has been doing Global Women Breakfast for the last couple of years, and Association of Chemistry Teachers India has been leading this program across India. And this year, more than 55 programs are happening across India this moment and today. So, and the purpose of doing this program of IUPAC is to have a kind of a networking, and that is the central key behind the program. And so that, we can discuss, the men and women both, about the important issues, related to women, particularly in science or even beyond science. So, and just to have a good networking and good groups for that. So this is the purpose of doing the program, and it goes very well. So thank you very much. And on behalf of Association of Chemistry Teachers, being a president of it, I welcome you for the talk. Thank you very much.
Thank you, sir. Now I request Dr. Saraugi to please go ahead with your address. Floor is all yours, ma'am. Ma'am, you are on mute mode, ma'am.
Sorry. Yeah. Sorry about that. Thank you very much, Dr. Kalpana Singh, Dr. Sneha, Dr. Pare, all of you, for the invitation and the kind words. It is my privilege and honor to be here and to be speaking to the students in one of the, top colleges, top government colleges in the country. So I think what I've planned is that I have planned, a few things. So since it's a global women's breakfast, I thought I will say a few things about my experience or my professional journey. And, you pointed out about all the difficulties that women and girls face, in science careers. So I thought maybe I'll speak a few minutes about that, and then I will go to the technical part of my talk. Would that be fine?
Yeah, sure, ma'am, sure.
Okay. So I'll share my slides. Are you able to see my slides? Ma'am?
Extremely sorry. Yeah, ma'am, we are able to see.
Okay, all right. So thank you very much. And so I thought, since it is about celebration of International Day of Women and Girls in Science, I thought I'll very briefly speak about my own journey. And I think there are many students, girls and boys in the audience who will be able to appreciate this. So I come from a normal middle class family, and actually the message I want to give first is that if I can do it, you can do it. Right. And just a little bit of my background. Basically, I came from a normal middle class family, and actually in my family, no one had studied science before me. Okay. And so I was the odd one. I was the different one, and I decided to pursue it because of my interests. But then, going forward, there was limited guidance because from the family. And but I still decided that I want to go ahead in the science career. And as mentioned, there are many difficulties and challenges that one faces, but one has to go ahead in something that they want to do. And so I decided to do that. Initially, I joined a college, Scottish Church College in Calcutta, where I did my bachelor's with honors in chemistry. And there I was very lucky to have very nice teachers and professors who guided me through this. And they said that you can go ahead and have a career in chemistry. So you can do a master's and you can do a PhD. If I wanted, I could do a PhD abroad in the United States or other top places in India. And in fact, at that time, I was not really aware of these opportunities, and they informed me and they counseled me through this, and I'm very grateful to them for that. So take the advice of your teachers and professors very carefully. So this is for the student audience. And if you really want to make it be successful in a science career, you have to work hard. But if you really want to do it, you can do it. And at the end of it, I feel that there's a lot of satisfaction in having a science career, where you can do research. You can do research on whatever you want, and you can do research on the top problems that maybe humanity, as a society, as a country, we are facing. And there's a lot of satisfaction in that. And so I wish you all the best. And what I would like to do now is to share some of the technical aspects of what we do using the chemistry expertise or the chemistry knowledge that we are gaining, and how the research scholars are involved, and how we take a question and we try to address it and take it forward. So my talk today is a combination of chemistry as well as some biology, because the problems that we look at are biology problems. So I will guide you slowly through that. So hopefully it will be clear. And then if you have doubts, so feel free to ask questions. And at the end, we will have some discussion as well. I hope there will be some time for discussion. All right. So before I go forward, I just want to show the picture of my research group. These are the people, some 20 odd people who have worked with me in the last 8 to 10 years in various capacities. Some of them are master's students, so they have to do a thesis. A master's thesis project, and then several of them are PhD students, and then some postdoctoral fellows as well. So these are the people who do the work, and our research is supported generously by IISER Bhopal and through SERB and the SERB Power Fellowship, and earlier I was a Ramanujan Fellow. So this is where the funds are coming from. So what I wanted to tell you today about is one story, one type of work that we do in the lab. So basically this is towards the development of small molecules, basically like potential drug candidates as inhibitors of protein aggregation. And since I think it's primarily a chemistry audience, so I've kept, molecule structures, etc. So you can appreciate, sort of the connection with chemistry here. So basically, first I'll state what the problem is. So you all know that in our body, as you are sitting here and listening to me, right, there are a lot of biomolecules in the body. So many chemical reactions are going on in your body, and most of these are caused by, are basically carried out by molecules called proteins, right? And so proteins react with other small molecules, other proteins, or other nucleic acids, DNA, RNA, etc., and carry out various functions in the body. And that's basically collectively, that's life, right, as we know it. Now, what happens is that proteins have specific three-dimensional shapes, okay? And that is necessary for their function. But sometimes what happens is that these proteins, these ribbons, you can imagine them as proteins, and they can come together, okay? So they can sort of clump together and form aggregates, okay? And when this happens, this is not very desirable, and then eventually they form like these kinds of aggregates, okay? So you can imagine like your hair, you know, when it's tangled up, then it becomes like knotted, and then it's not nice anymore. And then, you know, so basically proteins become tangled and then they are not functional. Okay. And this results in several diseases. Many of them you might be knowing the name: Alzheimer's disease, Parkinson's disease. All of these diseases are caused by protein molecules aggregating or clumping together. Okay, and it, of course, it has you know, problems, so it gives diseases, and then people have to be treated for the diseases. So one such protein that aggregates is insulin. So insulin is a protein in our body, and it helps to maintain the glucose concentration. So when we eat food, right, insulin is released, and then that insulin reacts with another protein called insulin. Basically, what happens is that it causes signaling, so the glucose is taken up by the body, okay, and then it is utilized for various functions, for like moving the muscle, etc. Now, one problem with insulin is that it aggregates. Just like I said, it clumps together into bunches and then makes these fibers structures. You can see that there are these long hair-like structures, and they become intertwined and entangled. So when this happens, the insulin is not functional, okay? And as now you may be knowing, many people who have diabetes, right, they have to be given insulin injection, okay? So basically the doctor prescribes them insulin injection, which they have to take. And so that can prevent basically the, so that the insulin can help for glucose uptake. But then sometimes this insulin forms fibrils, especially because when people are taking the insulin injection, the insulin is very concentrated. So that time it facilitates fiber formation. Okay, and then once this fiber is formed, this insulin is basically not useful. So that is bad for the patient. So basically we wanted to, you know, in my lab, we wanted to do something about this problem. As you know, diabetes has become a big problem in this country, worldwide, but also in our country. As you know, people are having, people's lifestyles are changing. And so diabetes is classified as one, you know, lifestyle disease, where basically, you know, so basically the point here is that there are lots of people in our country who have diabetes, leading to obesity and lots of other problems. So all of these people, you know, when they are taking these insulin injections, they are at the risk of being exposed to these fibrils, and then the insulin is not functional, right? So we thought, so they have to take basically more insulin injections. The dosage has to be increased. So you have to keep going to the doctor, and the doctor will increase the dosage. So we wanted to do something about this problem, okay, in my lab. So what we decided to do is that we decided to come up with small molecules, potential drug molecules, right, that can try to prevent this aggregation of insulin. So how do we go about this, right? So I've stated the problem, but the point is that as a chemist, how to approach this problem? That is the first thing, right? How we have a problem, how do we approach the solution? So what I'm showing you here is the structure of insulin. So insulin is a protein, which means it's a polyamide, okay? So you would have learned in your courses that proteins are made up of alpha amino acids, okay? So this one letter, each letter here gives the name of the amino acid residue that is there. So C stands for cysteine, S stands for serine, I stands for isoleucine, and so forth. So there are 20 of these that proteins are usually made of. So this particular sequence, when it is present, okay, so this is a protein called insulin, and this SS is for disulfide bonds. So there are these 3 disulfide bonds present. Now in this molecule of insulin, so I've highlighted here in these black ovals. So these are some of the residues which are important in this aggregation process that I told you about for the fiber formation. Okay. So what you can see here is that many of these residues, if you look at this, this is a leucine. Leucine basically has a secondary butyl side chain in the alpha amino acid, the R group, right, has secondary butyl. Valine is an isopropyl group, and then you have alanine, which is a methyl group, and then leucine and valine and so forth. So what I'm trying to highlight over here is that the parts that get aggregated have a lot of hydrophobic functionality. Okay, so those of you who are studying, organic courses, you would have studied, like, which groups are more hydrophobic, etc. So over here, this region that tends to aggregate are the region which are highly hydrophobic. So we thought what is happening is that these hydrophobic portions of the molecule, now think about the protein insulin, it is present in water, right? So these hydrophobic regions tend to repel water, so they try to come together and clump and then eventually form the aggregate, right? And you probably in some of your lab experiments, you may have seen, when things aggregate, they form a hazy solution and they precipitate sometimes. So we thought, well, okay, what is happening here is that hydrophobic molecules are interacting with each other and making insulin aggregate. Okay, so now if we can make small molecules, basically organic molecules through synthesis, that have some hydrophobic functionality, okay, then what will happen is we put them in this mixture, right, in solution. The hydrophobic regions will interact with some of these hydrophobic regions so that another insulin molecule will not be able to come together and aggregate. So we will inhibit aggregation. Basically, this was the hypothesis. Okay, so what we did was we made a bunch of molecules. So you can see the molecules are quite simple. So this is the pyridine core. And then we have some substitutions. Here there's a nitro group, carboxylic acid. And then here we have R1. So R1 we changed to isopropyl or isobutyl because, like I said, so there is leucine and valine, so which are similar to this. And we have some other side chains as well. So what we did was we made these molecules in the lab. Okay. Through basically amide coupling. You can see that these are basically, you take a aromatic acid, okay, like a benzoic acid derivative, right, or a pyridine derivative, and then you take the corresponding amine and then you condense them to form the amide. So this is how we made the molecules, and we tested whether they can inhibit insulin aggregation. Okay, so how do we test? So basically the way we test is this. We have a dye, okay, it's called THT or thioflavin T. That's an organic dye. And essentially what that dye does is it, you put it in solution and we put the protein. So as the insulin, this is the insulin structure that we just saw, as the insulin molecules come together and aggregate, the dye becomes fluorescent and we can monitor that signal, okay. So over time, initially there is no fiber, so there is no signal. Now fiber starts forming over here, okay, and eventually fiber formation is completed, so the signal saturates. So this is a readout of fiber formation, okay, using the thioflavin T dye. So that's what we did. And now we tested each one of these molecules separately, and I'm only showing you data for this molecule, which is M1, which is shown over here. So when we add M1 molecule to insulin, you can see that this aggregation is much less. So initially it takes a lot more time to start aggregating. And then as the aggregate starts to form, the THT signal increases, but it only increases up to this point, not all the way here. Okay, so we are having a significant reduction in aggregation. So that's something we wanted. So we're very excited about this result. And we also viewed this under transmission electron microscopy. So that's a way for us to image what's going on, because these molecules are very small, so we can't see much by naked eye. So what we can see in this case, when there is no molecule present, we have these network of fibers, which means insulin is aggregating. However, when we have the molecule M1, you can see clearly very, very low abundance of the fibrils. Okay, so this again means that our molecule M1 is able to inhibit insulin aggregation quite significantly. Okay, so that's where we started out. Okay, very simple chemistry concept that if we have hydrophobic groups over here, then that will prevent the aggregation of the insulin molecules, the self-assembly of the insulin molecules. Okay, so this was a very exciting result, but then what we wanted is this should be a flat line. There should be no aggregation at all. Okay, so we took these molecules and we started making derivatives of these molecules to see whether we can find such a molecule. So we made lots of molecules. I'm just showing you a brief list. So don't get overwhelmed by the list, but you just have to see that again, these are these, pyridyl amides basically. So we made longer versions. So here we have 3 pyridine rings. Earlier one had 2. And then we also connected, different molecules and we had different functionality and we tested them. Finally, we found one molecule called BPAD2. So basically it is this molecule. So here we have these pyridyl amides connected through a polyethylene glycol linker. And what we found was that, again, this molecule was much better than our earlier molecule M1 that I just showed. Okay, but still it was not inhibiting completely. So that was still a good result, but we wanted to get better. We wanted a molecule that will completely inhibit insulin aggregation, like I discussed. So we went and read some papers. And basically what we found, there were several reports that organic molecules containing sulfonate groups seem to be very effective at insulin aggregation inhibition. So that's what we wanted to do. So there are 2 molecules I'm showing you examples here. So these were good at insulin aggregation inhibition, and somehow the presence of the sulfonate moiety seemed to be important. So we thought, okay, let's take our molecule. This is basically the M1 scaffold that I first showed you. And then through an amide linkage, we made this sulfonate derivative. Okay, so we call it pyridyl amide dimer with a sulfonate. So we abbreviate it as PADS. So PADS is the molecule we made. And this was again made through quick condensation steps. So I'm not going to talk very much about the synthesis. Okay, so basically we activate, so here the acid, we make an activated ester, and then we have a nucleophilic attack through this amine group, and then we get the PADS molecule. Now, once we have the molecule, we wanted to test and see whether it can inhibit insulin aggregation or how well it can do so. So what we did was, we did the same THT assay that I had described earlier. Okay, now here you can see in black is the aggregation of insulin that I had shown you earlier. Now what we are doing is, excuse me, we are adding the PADS molecule in increasing concentrations. So here is the ratio, insulin is to PADS. So 10% PADS, then 50% PADS, and more. So this is 100% PADS, like basically equal, stoichiometrically equal amount of insulin and PADS. And what we see, the green line, okay, is basically a flat line. So you can't see, actually it's hidden behind the orange. Okay, so it's pretty much a flat line, which means PADS completely inhibits insulin aggregation. So this is something that we were looking for, right? So we're very excited about this result, right? And then at higher concentrations, obviously that is same. So if you add excess PADS over insulin in 2.5 molar excess, 2.5 fold excess, I'm sorry, then also you get the inhibition. So this was a good result, and then we thought, okay, fine, let's also do some imaging and find out. So here what we are showing, what we're doing here is, in this data, this is called circular dichroism, and essentially circular dichroism is a method to study the secondary structure of the protein. You know, proteins, I told you they are folded, they have specific shapes, and this measures the secondary structure. So in green is the normal insulin, the normal soluble functional insulin that we want. Okay, so that is giving what is called an alpha helical signal. When the insulin aggregates, it gives a beta sheet. So that signal is shown in this black line. Okay, so these signals are very characteristic of the shapes of the protein. So that's useful for us for interpretation. Now, when we put insulin is to PADS in 1:1 stoichiometry, we are getting most of this signal back. So you can see it's very much like the green line. Okay, which means that the insulin is properly folded and it is not clumped together, because then it would give this black signal. Okay, and we've also done imaging and like I showed you, so this is the insulin aggregate by itself. When we have equimolar amount of PADS, then almost no aggregates are formed, but very one or two, very little. Okay, so this is very good. Basically, this is what we were after. So we're very excited about this result. And here I'm showing you just a visual, because always the image is much more compelling than the data. So here is the insulin by itself. When it aggregates, it makes this hazy precipitate. You can see that there's a hazy precipitate here. But when we keep the same tube, we add PADS to it for 24 hours, there is no haziness at all. The tube is completely clear. Okay, what that means is there is no precipitation over here. So this is just a visual reconfirmation from whatever we were getting from the data. And you can also do it through, confirm this through NMR. So when you take the protein NMR, this is the protein NMR before the start of the experiment. So the protein is soluble and it is giving these signals. And when you add PADAS, keep it for 24 hours and then take the NMR again. All the peaks are retained. Okay. If the insulin had aggregated like this, it makes a hazy precipitate. So then that will just sit at the bottom of the tube. It will not give any signal. So we'll get a flat line. Okay. So this is another reconfirmation that insulin is in the soluble form. So then we ask, okay, so insulin, PADAS is able to keep insulin in the soluble form, but how does it affect the cellular toxicity? So the insulin fibrils are toxic to the cell. Okay, so we asked if PADAS prevents the fiber formation, then it should also prevent the toxicity. So that is the experiment we did. So this is more of a biological experiment. So here what we did was we checked the cell growth, the cell viability of the normal human cells. These are HEK cells. So basically the first lane is just the control. So this is a normal cell viability in the absence of any additives. When we put native insulin, the cell viability is maintained. We did this experiment in blue where only PADAS is put to check whether PADAS itself is toxic, because even if it is helping to prevent insulin aggregation, if the PADAS is toxic, then we can't use it for humans, right? So we found that PADAS by itself was not toxic. The cell growth was pretty much similar to the control. The aggregated insulin is in black, so that is toxic. So what happens is the cell viability decreases. Okay, so only about 50% of the cells are able to grow when you have the insulin fibrils. So this is consistent with the toxicity of insulin fibrils. Now, when we add insulin that has been treated with PADAS, so we saw that there was no fibril formation, and you can see indeed that the amyloid cytotoxicity is also gone because PADAS is preventing the formation of the amyloid fibrils. Okay, so this was a very good result. This is telling us that the toxic effects of the insulin fibrils are being prevented by our molecule PADAS. Okay, so that was a very nice result. So how much time do I have? Let's try to see. So then we asked, okay, how does the PADAS work? Right. So always we want to know what is the mechanism that is happening. So like I said, so these are the insulin protein. It has these various structures. So 6 molecules can come together to form a hexamer, or 2 to form a dimer, and so forth. And then it's the monomer of insulin that is the functional form, which helps in the glucose uptake and helps with managing diabetes, right? So basically, this monomer is the biologically active conformation. So that is the one that either binds the receptor, but if bad things happen, then that's the one that forms a partially folded intermediate and then goes to this fibril form that I was talking to you about, the aggregate form. That is the undesirable form. So we know that this aggregation is being inhibited, but how does PADAS act? What does it do? So what we did was, we took PADAS and we started chopping the molecule. So basically now we want to know whether all of PADAS is required or only part of it is enough for activity. So this is the full PADAS molecule. Then what we did was we just took this sulfonate group, because I told you that the sulfonate group is important. So we want to test whether sulfonate itself is good enough for aggregation inhibition. If that is the case, then we don't require all this aromatic region. So we made one of that molecule, another molecule where we took out the sulfonate and we just kept that, which I've shown you earlier. And then we took half of the molecule because there were these 2 pyridine rings. So we wanted to ask whether one is good enough, right? Because you don't want to have any extra stuff unnecessarily if it is not required. So here is the data for the THT assay that I earlier showed you again. So this is in black is the insulin by itself. Okay. And then what we found was this propionate, the sulfonate molecule, that was not able to inhibit insulin aggregation. When we have this PAS, which is half of PADAS, that works, but not so great. Okay, it inhibits to a little small extent. Okay, PAD, which is this molecule, does not seem to inhibit much at all, but PADAS completely inhibits, like I told you. What that means is, the whole molecule PADAS is required for full activity, because the sulfonate molecule is interacting with the insulin, as well as these hydrophobic side chains. So I told you about the isopropyl and the isobutyl side chains, right? So they are all required for activity, for binding to the insulin molecule. So we did a binding assay. Basically, here we looked at tyrosine fluorescence. Okay, tyrosine is a did a molecular docking. Basically, what we do is we take the protein. So these things that you see are the protein residues. Okay, and I'll show you this picture. Remember this picture of the protein that I showed you earlier. Okay, so this is just a three-dimensional arrangement of these, and these are what happens is one of the big problems in once the amyloid is formed, it is extremely stable. Okay, so it cannot be actually taken back because that's a thermodynamically very stable state for the protein. So, so disintegration of fibrils, any protein fibrils, is a challenge in the scientific community. What we found was that, in fact, using our molecule PADS, okay, it's able to disintegrate. So here we have done the same THT assay. If you have insulin by itself, nothing happens; it just stays there. But if you take the insulin fibrils and add different concentrations of PADS, over time, okay, you see the signal going down, which means that the fibrils are disintegrating. Okay, this is an extremely exciting result. Just hold on. So, yeah, biosimilars. Basically, these are variants of insulin.
Ishu ma'am,
yeah.
Excuse me. Madam, your screen is actually not visible. Actually, it has screen saving is, I think, screen sharing is just stopped.
It's stopped. Okay, let me try again. Sorry, actually. Were you able to see earlier?
No, it just went off 2 minutes.
Just went off. Okay, let me just
Nitesh Prajapat, please, you just mute your video because there are internet disturbances.
Is it visible now?
Yeah, now it's visible.
Okay, great. So basically now, once we showed that, we are able to prevent the aggregation of insulin and also disaggregate insulin, we asked, okay, the insulin that people are using, actual patients are using this. It's called insulin variant. It's called Lispro and Glargine. Okay, there are two variants. And essentially the only difference is that there are some side chains. The insulin amino acid residues are slightly different and they are marked over here, but the rest of it is quite same. So since these are the things, these are the products that actual patients are taking, for diabetes treatment, we asked, okay, can our molecule PADS inhibit these, okay, aggregation of these things? So we did that assay. So I'm showing you for Lispro. So basically this is the aggregation of Lispro by itself. And you can see that PADS at 1:1 concentration was able to also inhibit PADS Lispro aggregation to a strong extent. And in fact, it was even better for this Glargine. Okay, so again, these are just variants of insulin. That's all we have to remember. And we also confirmed this using circular dichroism, where the secondary structure, we have confirmed that it is able to maintain. Okay, so this is my last slide. So here I want to spend some time. So basically the point was, we showed that when we treat insulin with PADS, then the aggregation is inhibited and cytotoxicity is prevented. But if we want to give this to a patient, right, insulin protein after treatment with PADS, it should be functional. Matlab, whatever its job is, right, it should be able to do. because it's not able to do its job, then it doesn't matter, right, if the insulin is aggregated or not. So it should be able to perform its function in the body, even after PADAS treatment. So that is what we asked. So what we did was, once the insulin, if the insulin is functional, okay, it does, it starts a signaling cascade, okay, and there is a protein called AKT, okay, which becomes phosphorylated, okay, and we can measure, once we treat the cells with the aggregated insulin or PADAS treated insulin, we can check whether the protein AKT is phosphorylated or not, because if it is phosphorylated, it indicates that the insulin is functional. Okay, so first what we did was we did an assay where these are just the cells, so you can see there's some amount of signal, which means that some phosphorylation is happening but not much. If you take insulin which is aggregated, which means it's not functional, then also we don't see much difference. If we take native insulin, which is the insulin that is functional, then it gives a strong signal, right? Compared to these, the signal is much stronger, which means that this insulin is functional. It is able to phosphorylate the AKT. When we take insulin that has been treated with PADAS, okay, and it's kept for 24 hours, we can still see that the still strong signal for AKT phosphorylation we are able to see. Okay, so what that means is that after treatment with PADAS, the insulin is still functional. Okay, so this is something that we are very excited about, and we are now talking with various biotech companies who are actually selling insulin. So we are trying to test if the molecule PADAS, if we treat the insulin already with that, then it will prevent the aggregation, and so that will give a better outcome for the diabetes patients, because they will have to the aggregation will not happen, so the dosage will not change. Okay, so they will so over time they can use the insulin for a lot longer. The insulin can be preserved, and so that will be something that will be useful for them. So I think with that, I will stop here, and I hope I was able to convey, the main points of the type of research we are doing. So there are other types of, similar work that we're also doing. So maybe there'll be another opportunity when I can visit your campus and tell you about it. And thank you for your attention. And finally, I just want to show a picture of my department. So this is the chemistry department at IISER Bhopal. Okay, so, I invite you. We are always looking for bright students, motivated students who want to do a PhD with us. So please check out our website, and procedure and everything, dates, everything will be given on the website. So from time to time, the portal will open up. So please apply. And if you have questions, feel free to drop me an email, and I'll try to answer you. Okay. Thank you very much. Thank you for the invitation. And I'll be happy to take any questions that you may have about this. Thank you, ma'am. I'm done.
Okay. So thank you very much, Ishu ma'am. Thank you very much for presenting not only your views, but also your research within the stipulated frame of time. And that is the very important thing for a scientist, that to say the whole thing in few words, that is the most important thing that you have done today. You have just explained your very nice and interesting research. It was related to diabetes and insulin, so it was of I can say that pertinent. It was a pertinent thing for we all, because we are struggling with these things nowadays. Society is actually struggling with this thing. Diabetes has now become. I may say that after some time it will be just an epidemic.
Yeah,
right.
The way, yeah. In India, I think it's going to be an epidemic. It's already reaching there, but pretty much, it's a big problem for us. We have a huge population. We have a big population, and we are probably going to outnumber all other countries in diabetes patients.
After, I think, 45 to 50 years, this is very, like, it really needs efforts to keep diabetes away from you, I think.
Yes.
Because it's coming to you, but if you are wise enough, if you have many, I should not say wise, if there are many type of things, lifestyle problems are there, many things are there. So diabetes has reached up to that, and where somehow in some member of our family, they are struggling with diabetes. And even in my institute, I am seeing that all our staff members who are above the age of, who are above the age of 50 years are somehow struggling with diabetes and all.
Yeah, that's absolutely correct. Yeah, in fact, even youngsters, because I think there's a lot of
exactly.
the again eating habits and a lot of sugary processed foods they are eating, and
Trans fats and all that, and even type 1 diabetes. Previously, we rarely used to encounter this type 1 diabetes, but nowadays type 1, type 2, every type of diabetes we are just coming across, and many important people who are into gymming and all that and exercise and all that, even they are acquiring this diabetes. So it really becomes a factor to concern that why this is happening and why this is all. So really you did a great job. As part of research as well as part of as a scientist to do this research, it was very important. So thank you very much for being with us. Thank you very much for being here. And I just expect that someday you may be able to visit our campus, because Bhopal is very near to us.
Absolutely.
Dr. Mary Garson has also joined from IUPAC. So I welcome her also. Ma'am, I welcome you, Dr. Garson. I welcome you also. So thank you very much, ma'am, Ishu Saraogi, ma'am, for being with us. Thank you.
Thank you. Thank you so much. It was a pleasure.
Yeah, thank you.
Yeah, Professor Mary Garson, most welcome.
Thank you.
On
Can
behalf
you hear me? Can you hear me?
Sorry, yeah. Most welcome, Professor Mary Garson. On behalf of the Association of Chemistry Teachers India, in the capacity of the President and Professor and Head, Department of Chemistry, Madhav Science College, Ujjain, India. It is centrally located in India, very center of India. And we have been organizing a Global Women workshop for the last 2-3 years. And yeah, and I understand, and we all know that you are the person, behind all these kind of things. And in India, last 2 years, there was very good participation. And this year also, across India, more than 55 institutes today celebrating Global Women Breakfast. Just now, a very good scientist, Ishu Saraogi, a very Indian scientist from a very premier institute, IISER, Bhopal, has spoken. And we have gone through all your contributions in chemistry. And we are lucky to have today that you are a person, who has been very instrumental in shaping the program across the globe. So most welcome, ma'am. And we are looking forward to hear you on this occasion. Kalpana Singh, Dr. Kalpana Singh, you want to say a few things?
Thank you, sir. Thank you very much for welcoming Professor Garson on behalf of college and Association of Chemistry. Sorry, it was on mute mode. So thank you very much, sir. Now I would just like to read a brief profile of Professor Mary Garson for all the students who are sitting over here. Professor Mary Garson is an emeritus professor. She is FRCS, FRACI, MA Natural Sciences and PhD in Organic Chemistry, School of Chemistry and Molecular Biosciences from University of Queensland, Australia. Her primary research interest lies in organic chemistry and bioactivity of natural products. She has held many important leadership positions within the Royal Australian Chemical Institute as the first female president of the Queensland branch from 1996 to 97, and as chair of the International Relations Committee 1996 to 2004. Her contributions within the International Union of Pure and Applied Chemistry, IUPAC, the global body representing chemistry, have included membership of Division 3, Organic and Biomolecular, since 2006, and as the first female president of that division. She is currently an elected member of IUPAC Bureau and have a responsibility for global planning of celebrations of the centenary IUPAC in 2019. Right now, she is looking into, as Brajesh Parekh sir told, that she is instrumental in organizing Global Women's Breakfast. We welcome you, ma'am, and it's a matter of honor to say that the Marine platform, I may pronounce wrong, ma'am, so please pardon me for that. The Marine platform, Mary Trigela Mary Garsona, is named in her honor. So it's a matter of pleasure and it's a matter of honor for us. We congratulate you, ma'am, for this, and we congratulate you for celebrating Global Women's Breakfast today and making all we women proud for being associated and being listening to you today. Welcome you, ma'am. Floor is all yours.
Thank you very much for that. And Dr. Singh, you did very well to pronounce the name of the flatworm, which is Merytigrella merrygarsoni, which means little female sea tiger. And although we don't have time to talk about it today, I trained as an organic chemist. But when I moved to Australia, I started to work in marine science. I started to apply the techniques of organic chemistry to marine science. And it was as a result of that this little flatworm was named after me. So just before we start, I just thought I'd remind you that I come from Australia. And if you've not seen it before, of course, we have our national emblem, which is a little koala bear. This is a small one, of course. I hope that sometime you have the opportunity to come. I would like to say that I'm disappointed. I'm sorry that I missed the previous presenter, Dr. Saraogi, because I would have enjoyed listening to her discussion. I was, in fact, talking to another group in India, and I've also been talking to a group in Kenya this afternoon, but we had some connection issues. And shortly I will talk with a group, if it works, a group in Tashkent, which is in Uzbekistan. So I'm learning a lot of geography this afternoon. Anyway, thank you for inviting me to interact with you today. So I'm going to tell you a little bit about how the Global Women's Breakfast event started, but I want to talk particularly about the future of chemistry and the role that young people have got to play in the future of chemistry. And when I say young people, of course, I mean both men and women. Obviously, because we're doing the Global Women's Breakfast, we're focusing on women chemists and male allies, and together thinking about what we've got to do to grow a better future. And I don't know whether you've watched yet the welcome video, the short video presentation that IUPAC put together, which has got some very inspirational words from the president of IUPAC, Javier Garcia Martinez from Spain. It's absolutely inspirational. And it also shows pictures of chemists from around the globe. And I did notice that there was a group of chemists in front of the Taj Mahal in Agra who were part of that. So if you haven't looked at it yet, perhaps do that later on this afternoon. Anyway, back in 2011, we held the International Year for Chemistry to celebrate the achievements and accomplishments of Madame Marie Curie, Nobel laureate in chemistry, also Nobel laureate in physics. And we wanted to network. And unwisely, I said to a colleague of mine, oh, women know how to network, they go out for breakfast. That is how this global networking activity was born. It was a simple statement I made to a colleague, and later on I realised that I would have to champion it. And in that first year, we had participants from over 40 different countries, including, I'd like to say, India. After 2011, I said, I'm not going to do this again. It was a lot of work. Let's have a rest. 2019, so 8 years later on, was the International Year of the Periodic Table. These are UNESCO flagged activities. And so we decided to relaunch our global networking breakfasts. And I co-convene that now along with Dr. Laura McConnell from the United States. And I need to tell you that all of the technical stuff, the website, all of the wonderful things that we put together are the brainchild of Laura. I don't do any of that technology stuff. So this is the 5th annual Global Women's Breakfast. And this one is particularly special because once again, because it's an international year, it's the International Year of Basic Science and Sustainable Development. And so we've joined together with other science unions, for example, the International Union of Physics, and other science academies to celebrate that. And we're thinking in particular about the United Nations Sustainable Development Goal, number 5, which is gender equity. In other words, empowering women and girls in science. So every year when we run this breakfast, we choose a date which is close to the International Day for Women and Girls in Science. And that date is February the 11th, which of course was last Saturday. I'm just stopping because I noticed that a couple of people have raised their hands. So I'm just wondering, I hope that can you let me know that you are hearing me okay?
Yeah, ma'am. We have resolved their issues, ma'am.
Sure. Okay, I'll just keep going. All right then. So February the 11th is usually that International Day, and that was on Saturday. And we always choose the Tuesday closest to that date. And of course, this year that meant it was Valentine's Day, February the 14th. So I hope that today we're all feeling the love for chemistry in our conversations. So what this is about is absolutely connecting, celebrating, and building a better, fairer, and more inclusive future for chemistry. So what we want to celebrate is the fact that today, thousands of people, we haven't done a count yet, we think that probably about 30,000 young people from around the world will be leaping out of bed to come and celebrate our global networking breakfast. I've seen some images of breakfast I just saw one from Malaysia, where there were clearly about 150 people in the room, which is a fantastic number. So thousands of people in hundreds of cities are joining together in our global conversation. And this year we've registered over 380 different events. I think the number actually might be 389. If you have time later in the day, please do follow our website. The website address is www.iupac.org/GWB. And you can see the global map and you can see the red hearts on it lighting up as breakfasts start up all over the globe. And breakfast is taking place in at least 75 different countries today. Since 2019, more than 100 different countries have joined in one or more events. This year, we've got participation from some new countries joining in the first time, including Cambodia, Vietnam, Burma, Myanmar in other words, in Africa, Cameroon and the Sudan, in the Caribbean, Honduras, and actually in Europe, Slovenia has joined in for the first time. So each year new countries join, and as was mentioned in the introduction, India has the largest number of breakfasts, and congratulations on that. Well done, India. India always has a competition with Nigeria to see which will host the most breakfasts. The Nigerian chemistry community is very strong, and they love to hold breakfast, and they have a lot of local activities and eat local foods and wear colourful costumes. So now I want to tell you about what I think the message is this year. And I don't know, I haven't seen analyses from India. I would be interested. But in my country of Australia, the number of women In fact, the number of young people doing STEM subjects, and by STEM I mean science, technology, engineering and maths, is not increasing, it's decreasing. And that means that many sectors of our society, the families around us out there, just in the public, are not very knowledgeable about science. They are STEM illiterate, really. And this is not a good thing. It's against a background where all of us, as we move into probably a challenging future, science is so important if we think about the needs of the global economy and sustainability. We've only got to think about requirements for clean water, food production, food security, safe medicines, cheap medicines. I know that India has a very important role to play in the production of medicines globally. And for example, that it was thanks to the introduction in India of manufacturing processes for making drugs against HIV and the AIDS and a lot of other antiviral drugs that the global cost of those medicines has decreased. So you have a really important part to play in that. New materials. I was listening this morning to a talk from Australia where we're getting some new submarines, apparently, nuclear submarines. And the defence people were telling us that we're going to need a lot of trained PhDs in both chemistry and physics who understand the interaction of radioactivity and nuclear fuels against these new materials if those submarines are to be effectively used in our waters. So that was quite a surprise to me. I talked also this morning with Taiwan, with China Taipei, with chemists from a beachside city outside of Taipei. And China Taipei is an important country worldwide for the production of semiconductors and chips. It has the largest manufacturing organization worldwide. A lot of semiconductors and chips are designed in the States, but the production facilities are mostly based in Taiwan. So there's a pressing need there for people with chemistry backgrounds, but of course with a good link and understanding of maths, physics, engineering and materials science. So this, if you like, is the role of the International Year for Basic Science and Sustainable Development, that we begin to join the dots together to think about the chemistry in the context of these other sciences. I have to say that my own training in Cambridge was a little bit traditional and strongly focused on organic chemistry. And when I moved to Australia and started to become interested in marine science, I discovered a whole new world of interesting science out there that I knew nothing about. I ended up working on marine sponges. And at the start of that research, I couldn't even have told you what a marine sponge was. It's in fact the lowest form of multicellular life. And I certainly didn't know how to identify them. Oh, and by the way, one of the more interesting challenges that I had when I went to Australia was that I had to learn to scuba dive. I couldn't really swim very well, but I learnt to scuba dive and to go underwater to collect my own samples. And of course, that's how I found that flatworm. So by telling you about the intersectionality of chemistry together with these other sciences, what we're really thinking about here, and it's an important message, is that modern day science is not about individuals, it's about collaborations, it's about teamwork. And there are lots of studies out there that show that diverse teams made up of talented men and talented women working together are more productive, they're more creative, but the working environment for those teams is happier. So teams that lack diversity tend to produce ideas and solutions to issues that tend to lack novelty. So in order to strengthen the science of the future and the contribution of chemistry to the future, we need to, particularly in those countries where participation is going backwards, we need to strengthen the participation of women in STEM. We need to strengthen the participation of all young people and to, as far as possible, encourage them to think about science as an intellectually challenging and personally rewarding and hopefully financially rewarding as well occupation. I mean, I probably shouldn't use the word, but to discourage them a little bit from thinking that they should do business studies or go into information technology. Because although I'm not able to talk to you in any detail about my science today, I'm passionate about the science that I've done, about the interconnection of organic chemistry and marine science. And I hope that all of the people listening to this presentation will find their chosen career path, identify a passion for science and take it forward. So we need to get the setting right for all scientists, young people. We need to, particularly where undergraduates, postgraduates, early career researchers, people doing postdoctoral research. I know that a lot of scientists from India choose to go overseas to do postdoctoral research, and it's important to bring them back into India to strengthen your scientific industries. So as I kind of indicated, we need to share the key messages that science opens the doors to career paths that are challenging, technically challenging. I explained to you that I had to learn to scuba dive. It was actually quite terrifying. And I also had to think about a lot of new science when I moved to Australia. But it was rewarding, and it's important not to be fearful about taking on those challenges and to dig in, to feel the motivation and just to stick at it. And the progress that collectively young people can make is going to be hugely beneficial to society in the way that I described. So the chemistry teacher, the science teacher profession has a really important part to play in this overall process of so that we can impact on young people into the future, make the very best use of their talents and skills, that's all of you, and impact on the future of chemistry. So I hope that in these few words, and I'm sorry that they are few words because I seem to have been talking all day from about, it's actually 6 p.m. here in Brisbane at the moment, and my day started at 4 a.m. when I had a conversation with the people who started, the breakfast started in New Zealand at about 4 a.m. and has been moving around the globe, as you will see. In fact, I'm going to finish my conversations about science and mentoring and leadership and encouraging young people into science and to think about making connections to other sciences at 11 p.m. tonight. That's in 5 hours' time, when I'll be talking to the International Younger Chemists Network, who've got a webinar, and I'm fortunately going to be able to show them a few pictures. I didn't organise that for you. So I seem to have been speaking all day on this message. I hope that in the conversation, I've seen your program, and so I hope that you have a valuable conversation together, both men and women. We look forward to hearing how the session goes. Please do send us some photographs. We will be sending out some emails later in the week asking you to just give us a little bit of information. Pictures speak more than words, don't they? And to let us know how you get on. So thank you so much for letting me join you, and have a great day. I'm going to hold up my breakfast mug, although really, because it's late.
Me too.
You have one too. Everybody has one of these. These of course come from there's an IUPAC shop. You can buy lots of other things, by the way. I've actually here got, this is my laptop holder. And there are also some pins.
Oh,
Things. These little lapel pins are quite good, actually.
right.
We've been giving them out as little prizes for people during the day. So anyway, please do take a look. And as I said before, please do have a look at the global map and enjoy watching as new countries join in the conversation. Thank you so much. Thank you for hosting these events in the past. And we hope to see you very much joining with us again into a future breakfast.
Thank you, ma'am.
Thank you. Can you hear me? Yeah. Yeah. Thank you, Professor Mary Carson. It's wonderful. Your words are very important to us, and particularly our younger generation and our students. And your contribution, your thorough contribution, the story is very inspiring. How you moved to Australia and all your these underwater kind of things, wonderful. And still you are so energetic and so motivating. So please keep motivating all the students across the globe, not only in Australia and New Zealand. So we are lucky to have you today for your discourse. Thank you very much. I understand. From morning 4 a.m. to 6 p.m. already, and you may have to speak for another 2-3 sessions. But thank you for accepting our invitation and keep guiding us always. Thank you so much.
Okay, thank you. And I will take my leave and say goodbye. Thank you so much.
Thank you very much.
Thank you.
Okay, bye.
Thank you.
Bye.
Professor Usha Nair, madam, has also joined. Madam, most welcome.
Thank you, sir. Thank you.
So we were listening to Dr. Mary Garson from IUPAC and Professor Emeritus, University of Queensland, Australia. Now we have with us another, I can say that we have with us another lady of substance, chemistry, quality. All of these things are synonymous with the name of Dr. Usha K. Nair. Whenever a thought comes in our mind about Usha ma'am, there is only one thought, that it should be quality, 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 and quality. She's a chemist by profession, and she's basically, initially she's a chemist, then she just took chemistry to another height of, I can say that, quality, and now she's practicing the quality in Madhya Pradesh and leading Madhya Pradesh towards the new arena of quality. Dr. Usha K. Nair is Professor of Chemistry. Presently, she is OSD Madhya Pradesh Madhya Pradesh Higher Education Quality Improvement Project, State Project Directorate, Department of Higher Education, Government of MP. She is Member Secretary, State Level NACSL, and Nodal Officer for Atmanirbhar Madhya Pradesh Output Indicator 3, NAC. She has a teaching experience of UG and PG classes of 38 years. She has specialized in physical chemistry, and her field of interest for research are synthesis of complexes and their physico-chemical analysis. Madam has undertaken many administrative assignments. She is in charge of different cells, Deputy Controller of Exam, Confidential Section, and she is a member NAC Steering Committee for the colleges, member secretary, state level NAC cell. She has done diploma in Sri Satya Sai Educare from Institute of Satya Sai Education, Thailand. She is an alumna and diploma holder, and I respect her as a person who is perpetuating and who is bringing in the quality in all the perspectives. Welcome you, ma'am, and I congratulate you on this Global Women's Breakfast. And we are just seeking your blessings, and we just want to listen to you.
Just one word for Madam. Meticulous working, flawless working, but at the same time, very polite. Thank you, ma'am. Most welcome.
On this great day, when we are celebrating the Global Women's Breakfast with women and girls in science, I extend my greetings to each one who has joined this meeting. I feel honored to be sharing a platform with a stalwart, Professor Mary Garson, and I was extremely happy to see that young scientist, Dr. Ishu, presenting her paper, presenting her a lecture, which was also very inspiring. And I extend my gratitude to Government Mother Science College for giving me this opportunity to be here, especially Dr. Kalpana Singh, whom I adore as a person who I think for meticulous working, I think that she should be given that award. And then Dr. Brijesh Pare, whom we feel proud to see as the president of the Union of Chemists, the group of chemists from all over India. We are really extremely happy that such stalwarts are making us proud in our state, representing chemistry and again quality by getting A++ grade for your institution. Your every contribution from both of you has gone a long way in getting A++ in the whole nation, the only government affiliated college to have got this A++ grade. It is really very heartening, and I'm extremely thankful to you for considering me for this Global Women's Breakfast program of this year. As Madam Garson already reminded,
Light.
are you able to hear me?
Yes.
Yes, ma'am.
Yes.
Yes, ma'am.
So as Madam Professor Garson highlighted, that there are 78 countries participating with more than 30,000 participants in more than 400 events that are being organized for this program. And we are also, celebrating the role of women and girls in science. So this makes me feel very proud that I also belong to this subject, which is my favorite subject, chemistry. And so before I say anything, I offer all the words that I am going to say to my mother. I dedicate it to my mother, who was also a professor of chemistry, a renowned professor of chemistry, who has inspired me throughout by her virtuous and knowledgeable career. She was an organic chemist, and she was my teacher also. So I offer my salutations at her feet before I begin. So women and girls can play a very vital role in ensuring the diversity in research, expanding the pool of talented researchers, and bringing in fresh perspective. Now, gender equality is a global priority for UNESCO, and therefore the support for young girls, their education and full ability to make their ideas heard are for development and peace building of our nation, of our country, of the world at large. Now, girls and women have to develop their mindset They have to learn from their failures. They have to be inspired by successful women, provide opportunities to succeed, help, and we have to help children, girls, to participate or to be part of STEM, that is science, technology. So Indian women scientists have, those who are playing a major role, have to come forward and address this issue. The girl students have to be guided and promoted to develop that scientific temper. In our country, which is very conservative, especially in our state, which is very conservative, still, they feel that girls must take arts, pursue arts, because ultimately they have to get married and they have to settle in a family. But today, looking to the present times, the present trend, the advancement of the technology, it is very imperative that we promote our girls to develop that scientific temper. Now, this scientific temper is what? It is that open mindset, ready to welcome new ideas, the new light, new knowledge, new experiments, wherever they are able to get it from. So that quest for new things and to go deep into it has to be nurtured and fostered for our children, for our students. We must have that logical, for scientific temper, they must have that logical and rational thinking, that reasoning and practical knowledge are very important. So what I feel is that the 4 pillars of education, we all know that it is learning to know, learning to do, learning to be, and learning to live. From that, learning to do is a very important aspect that we must concentrate on so that our children, our students, the girls develop that aptitude for science. As I have been thinking about this topic since Dr. Kalpana Singh informed me about it, I felt that whether it is science or arts, both have a bit of each other in each other. There is science in arts and arts in science, because science also needs a lot of imagination, a lot of creativity, along with reasoning and practical knowledge. And so is this subject called chemistry so very important in our lives. From the time we are born, in day-to-day life, we have been seeing that everything, where it is the use of chemistry. And science, technology, engineering, and mathematics, which we call as STEM, have many choices for women also, yet they are underrepresented in these fields. And that is what we have to address through this Global Women's Breakfast program. Women have played very important roles in STEM, but they tend to be marginalized, saying that it is not their cup of tea. There are very few Nobel laureates women amongst women. There are very few people who have senior research roles amongst women, and that is where we have to concentrate and see to it that our students develop that aptitude, that liking for science, chemistry, and so that they can achieve greater heights in the field of whether it is research or teaching or whatever endeavor or an entrepreneurship. They have to have that liking for chemistry or for science as a whole. So it is imperative that we provide all possible avenues for girls to pursue their qualities of curiosity and intuition. These two fields have to be encashed and, enhanced in our children. Our education system must encourage girls and women to undertake, develop their skills and allow them to unleash their capability for which they have been created by the Almighty. For that, they need to be given accessibility to education, affordability, equal opportunities in whatever and all required possible the exposure to science inside them. Since they are endowed naturally with qualities like patience and innate nature of love and empathy, affectionate behavior coupled with knowledge, skill, critical thinking, problem solving, creativity, and such qualities. The research aptitude in our children needs to be identified at a very young age, and all conducive environment must be made available to pursue their passion. There are many people, there are many children who have tremendous capabilities, tremendous potentialities, tremendous talent, but they are not able to bring it out due to the barriers that which we are talking about today. And those are the barriers which need to be broken and they be allowed to excel in whatever fields they want to. Wherever they are placed, the women and girls, wherever they are placed, history tells us, history has been, has validated this fact time and again that they have excelled in all fields of life. In fact, the greatest validation comes from the Almighty, who has chosen them to be the vehicle for creation, the motherhood, which again validates the hidden enormous role of women and girls. The enormous untapped talents need to be unleashed, starting with filling the classrooms. Children have to be brought to the classrooms, laboratories, and boardrooms with women scientists. We have to fill these places with women scientists. That should be our effort because that is where we have to bring in quality, for which I have always been saying that our state is slightly backward in doing research activities. And that is where we have to bring our children to the laboratories. We have to fill the classrooms, and we have to make them do these experiments in science so that they develop that aptitude. With women and children, women and girls, as being myself, let me take my example, being a student of science, especially chemistry, it has helped me to, not only the subject in academics, but it has helped me to develop discipline, memory power, observation, doing things myself to understand the nitty-gritties of it, a love for experimenting, analyzing, applying the knowledge for making something new. That has been my observation about myself being a student of science. Being a woman, I have been able to successfully transfer all these qualities to my better half and children, also to give them insights into areas which I could not achieve. Also to provide a better family life with a scientific approach, whether it be academics, whether it be food, whether it be nutrition, whether it be health, housekeeping, cleanliness, safety measures, and above all, spirituality. All these things I could transfer from me to my family only because I am a woman, I'm a girl. I have been a girl science student. So only if you educate a girl, if you give education to a girl, we can be assured that we have given education to a whole family, because she is going to transfer that to the whole family. And if she is a student of science, then all these aspects that I said, whether it is academics or food or health or nutrition, or it is cleanliness, or it is safety and security, all will be taken care of by that person. And ultimately, I said spirituality, because it is spirituality which gives you that inner strength to develop that inner potentialities, that intuition power for creativity, for imagination, to do greater things in life, especially in the field of science. Only when this spiritual inner strength can be used in science for the service of mankind, can we do meaningful research, ultimately aimed at benefiting the upliftment of humanity and to bring peace in the world, to be guided by the universal values of truth, right conduct, peace, love, and non-violence. Because let me tell you, the emerging trends of chemistry, whether it is sustainable chemical production, artificial intelligence as a predictor of viruses, improved efficiency for personalized drug treatments, where the creation of wearable sensors for healthcare monitoring, or it is blue hydrogen or green chemistry or nano particles, whether it is climate change or water and food production, any field of chemistry would require these 5 universal values, which will ultimately lead to the peace for the whole world and benefit the humanity at large. So I would end this small piece of talk that I have been requested by Madhav Science College. I would end this with the saying that when there is righteousness in the heart, there will be beauty in the character. When there is beauty in the character, there will be harmony in the home. When there is harmony in the home, there will be order in the nation. And when there is order in the nation, there will be peace in the world. So let us all, as students of science and especially chemistry, contribute to bringing in our next generation, our children, to love this subject, to pursue careers in science, and to undertake active research which will benefit the entire humanity, as we have seen in the COVID times, where the entire science fraternity came together to develop innumerable techniques and technology to help humanity to come out of that this disaster. So with these words, I express my gratitude to the entire team of Madhav Science College for having me for this Global Women's Breakfast. And I also feel extremely proud to be a women scientist, a chemist, to participate in this program. Thank you very much. My best wishes to each one of you who is participating in this, and thanks to each one of you for hearing me patiently. Thank you.
Thank you very much, ma'am. Thank you very much for sparing your valuable time. We know that how busy you are and how much involved you are in the NAC accreditation processes of this state. So I, with my colleague Sneha, who is just been an instrument in organizing this Global Women's Breakfast, I thank you, ma'am. And with this, I invite Dr. Brajesh Pare. So please say a few words for this program.
Thank you. I'm audible. Yeah. Thank you, Madam Dr. Usha Nair. Thank you for accepting the invitation. And you spoke wonderfully. We are expecting the same. And Yeah, we understand your spiritual inclination also with the work. That is very easily exhibited and can be seen, while you are working, and what you spoke, that is all in line with the way you have been working for such a huge project in Madhya Pradesh for bringing, more number of colleges for NAC. And I understand, that how difficult task it is, because it's not only the administrative work, but it requires a lot of motivation also. And that is what you have been doing in addition to your administrative responsibilities, because, for going for a big task, you need to motivate your, the workforce, rather, I would say, the professors and the principals of the colleges, that you have been already doing, and you spoke very well, and that is very encouraging for all our students. And keep blessing us, ma'am, and keep guiding us. Thank you very much. And I am thankful to all the three speakers, Professor Ishu, and then Professor Mary, and Dr. Usha Nair, madam. Kalpana Singh, madam, thank you very much for organizing it very nicely. And thank you very much to all of you, to Professor Arpan Bharadwaj, our additional director, and our principal, Dr. V.K. Gupta, sir, for their constant encouragement for such events. Thank you very much.
Thank you very much, sir. I once again thank all the speakers, Professor Usha Nair, madam, Professor Mary Garson, Dr. Ishu Saraugi, Additional Director Ujjain Division, Professor Dr. Arpan Bharadwaj, Principal Dr. V.K. Gupta, Head of the Department, and ACT Chairperson Dr. Brajesh Pareji. With that, I am very thankful to the IUPAC also, who provided us, and ACP also, who provided us this opportunity to go ahead and to organize this Global Women's Breakfast. I am thankful to Sneha, Swati, and all my students who are here with us, PG students. They have joined us on this Google Meet as well as on YouTube. They are just going through this program live. So, sir, ma'am, thank you very much for being with us. Thank you very much. And once again,
Thank you. So nice of you, please. So nice of you. Thank you very much. Thank you, Dr. Kalpana Singh, ma'am.
Yes, sir. So I request all the students of Mother Science College who are listening it from this program from campus to come forward in front of conference hall for a photo.
Yeah, with our periodic table wall.
Yes.
Thank you, Madam Dr. Kalpana Singh. And yeah, it was really successful. Both, all the three speakers, they attended nicely. And thank you to all our students also.
Okay, you can leave the meeting now. Thank you very much.
चलते हैं बच्चों को बुला के लाते हैं सर प्लीज एक फोटोग्राफ बच्चों को बुला के लाओ यस मैं डिस्कनेक्ट कर दूं